Okay, this slide was presented in the first lesson and is presented to refresh the feature of the resting state EEG activity in the condition of eyes closed and eyes open. This was the trace of EEG activity showed by Hans Berger in the first recordings in humans about 1 century ago. As you remember, the EEG activity during the resting state, eyes closed, show high amplitude in a frequency from 8 to 10 Hertz generally. And when there is the opening of the eyes, the alpha oscillation disappear, and an oscillation at the higher frequencies from 20 to 30 Hertz is visible. And you see in the middle of the picture that the EEG oscillations show a frequency around 20 Hertz during eyes open. This is the reduction of alpha oscillations from eyes closed to eyes open as a reflection of a cortical activation of visual and spatial neural systems during eyes opening. What are the generators in the brain of resting state alpha RINs? Vincenzo Crunelli is an Italian researcher working in Cardiff in United Kingdom, and his group was able to provide some very nice findings about the involvement of the cerebral cortex and the thalamus in the generation of ongoing alpha RINs in quiet vigilance. In the left part of the picture, you see a model of the cerebral cortex and thalamus. Particularly, you see the cortical pyramidal neurons in the cerebral cortex and thalamocortical neurons in the thalamus, particularly the relay thalamic nuclei of visual system, the lateral geniculate nucleus. You see that in the model, the cortical pyramidal neurons show axons modulating the activity of the thalamic relay nucleus of a visual system, the lateral geniculate nucleus, and also of neurons of nucleus reticularis thalamus. On the other hand, the thalamocortical neurons have axons modulating the activity of cortical pyramidal neurons. So there is a reciprocal connectivity between cortical pyramidal neurons and thalamocortical neurons. Vincenzo Crunelli implanted electrodes in the cerebral cortex and thalamus of cats in behavioral cats and also in tissue preparations in vitro. And in both cases, he was able to record oscillations in the local field potentials at the cortical and thalamic level, showing this kind of oscillations. You see in the middle of the picture, and please pay attention to the time scale of 500 milliseconds. And you see that if you count the peaks of these oscillations of local field potentials, they show the frequencies of alpha rhythms around 10 Hz. And indeed, you see that in the graph of power density spectrum in the right side of the picture, you see the EEG power spectrum from cortical electrodes in red and the lateral geniculate nucleus in black. And so you see that the maximum power of the local field potentials is around 10 Hz. So this was a very nice demonstration that the scalp recorded resting state EEG alpha rhythms are generated by the synchronization at alpha frequencies of cortical pyramidal neurons and thalamic neurons, and that they are connected each other, forming the so-called thalamo-cortical circuits and corticothalamic circuits. So the idea is that these thalamocortical and corticothalamic circuits oscillates as an activity at around 10 Hz and provide a sort of idling state of the cerebral cortex, a relaxation, a sort of cortical inhibition of the mode of quiet vigilance. The group of Vincenzo Crunelli was able also to establish an association between the peaks of alpha rhythms in the local field potentials recorded from the thalamus or cerebral cortex and the spikes of the single neurons of the cerebral cortex and thalamus. And you see the results in the picture. In the picture, each action potential is represented by a blue dot. And so you see that when you consider the times, the axis of time, which is horizontal, you see that there is a concentration of spiking of the neurons in certain short periods. And the period with the concentration of spikes is followed by a period of short silence of the neurons. So you have a sort of burst of activity represented by the spikes, and then a short period of silence, and then a short period of spiking, and then a short period of silence, and so on. And this demonstrates that there is a strict relation between the time curves of the alpha oscillations in the cerebral cortex and thalamus and some short periods of high frequency of spiking of the neurons. So alpha rims are not an epiphenomenon, are not something unrelated with the activity of the neurons as revealed by spikes. You see that I use the traffic light in red in relation to alpha rims because red in the traffic lights means stop, inhibition, not to go. And we can interpret the resting state, eyes closed, ample alpha oscillations in the EEG recordings as a measure of the ability of the brain to establish a sort of low cortical activation, a control of cortical excitation associated with a low vigilance, quiet vigilance, not selective information processing from sensory channels or autobiographic memory or semantic memory or motor systems. Another important scientist, Fernando Lopes da Silva, shows some interesting findings of local field potentials recorded from visual cortex of dogs to demonstrate the localization of the cortical sources of EEG activity. In the picture, you see in the center of the picture representation of a cerebral cortex with the layers of cerebral cortex and one electrode implanted perpendicularly with the surface of cerebral cortex. And you can see the electrode contacts recording local field potentials also in the right side of the picture. They are the black rectangles. From these contacts, Fernando Lopes da Silva's group was able to record local field potentials. In each contact, you have a local field potential resembling the ongoing EEG. The difference between this local field potential and EEG is that the pin of the electrode is very small in this intracerebral recordings. With an electrode within the cerebral cortex is very small, the tip of the electrode, and so you can capture the electrophysiological activity of a small number of cortical neurons. When you record scalp EEG, you have a large cap electrodes with several millimeters of diameter, and you record from millions and millions of neurons. But you can see that the local field potentials show clearly an alpha oscillatory activity. You can see the time scale horizontally, 250 milliseconds, and you can count the peaks of the local field potentials around 8, 10 Hz alpha rims. Even in this case, the dog was not involved in any motor or sensory tasks, and so is a sort of passive wakefulness. The red light in the traffic light means this kind of cortical relaxation and no task-related cortical activity. And so the observation of Fernando Lopez da Silva was that the source of these local field potentials were about in the middle of the cerebral cortex. Module. In other words, you say, okay, you see the layers of the cerebral cortex from the surface layers to the deep layers. You see this EEG alpha, the local field potentials showing alpha rims. But if you follow the vertical dotted line, you see that in the surface electrode contacts, the dotted line indicate upper peaks of the local field potentials, and the lower contacts at the same time of the vertical line shows the lower peaks. So in other words, you see that the higher peaks and the lower peaks are reversed in polarity. And this reversal of the polarity implies a dipolar cortical source in the middle. And so he put about in the fourth layer of the cerebral cortex the source of alpha rims. Another important contribution to understand the sources of alpha rims by Fernando Lopez da Silva was reported in this slide. Again, he recorded from the cerebral cortex of dogs and the thalamic nuclei from dogs simultaneously. And so you see that the picture shows the local field potential recorded from the visual cortex of dog, and 10 is the number of the electrode. In the middle, you see a local field potential recorded from lateral geniculate nucleus, the visual sensory thalamus. And in the bottom of the picture, you see the electrode 9 implanted in the visual cortex. So electrode 10 and electrode 9 are located in the visual cortex of a dog. You see that the oscillations of local field potential, you see the 1 second of the time scale. The shape of the oscillations in the 3 electrodes look like very similar. suggesting that there is a functional connectivity and interrelatedness between the local field potentials in the three electrodes, cortical and thalamic. And indeed, when you see the power density spectrum of local field potentials from electrode 10, visual cortex 10, you see that the maximum power of the oscillatory activity is at 10 Hz. When you see the maximum power in the EEG power spectrum of the local field potentials from lateral geniculate nucleus, you see that again you have that maximum power is at 10 Hz, like the visual cortex electrode 10. And the same is true for visual electrode 9. Another way to represent the local field potentials is to compute the spectral coherence spectrum. In the spectral coherence spectrum, you see that in the vertical axis, you have the coherence, the correlation, the similarities of the phases of the oscillations. And in the horizontal axis, you have the frequencies. So the spectral coherence between the local field potentials in two electrodes is represented in the upper graph for visual cortex electrode 10 and visual cortex electrode 9. The two waves of a local field potential were represented in terms of coherence in the graph. And you see that the maximum coherence, the maximum interrelatedness of the phases of the oscillations, the maximum correlation between the two local field potentials were observed at 10 Hz. There is another peak around 20 Hz also, but the higher coherence is at 10 Hz. So this would suggest a functional connectivity between the local field potentials recorded in near places of the cerebral cortex in the visual system. In the middle, you have a graph showing the spectral coherence of local field potentials recorded from visual cortex electrode 10 and electrode in lateral geniculate nucleus of visual thalamus. And you see again that there is a lower but substantial peak of spectral coherence at 10 Hz. And this shows that, again, there is a functional connectivity between thalamocortical neurons and cortical neurons and the generation of alpha rims. So we observed in these two slides a network of thalamic and cortical neurons which produce alpha rims in a very interrelated way. In another study, Fernando Lopes da Silva wanted to test the hypothesis of a strict correlation between scalp recorded EEG alpha rhythms and the activity of the cerebral cortex and the thalamus, and used a combination of resting state EEG activity and resting state bold activity recorded by magnetic resonance imaging. As you know, the bold signal of functional magnetic resonance imaging can be obtained by the T2 star sequences during the recording. And this particular sequence of magnetic resonance imaging is sensitive to the ratio between oxygenate hemoglobin and deoxygenate hemoglobin. In other words, when there is an increase of oxygenated blood in a specific place of the cerebral cortex, you have a change in the ratio between the oxygenated hemoglobin blood, an increase when compared to the part of the blood which is poor of oxygenated hemoglobin. So when you are active in the cerebral cortex, for example, you have a big change of this ratio, and so you have a big, bold signal showing cortical activity. And the same is true for subcortical structures. And this is typically represented by colors in the magnetic resonance imaging. In this picture, you see the two sagittal slices of functional magnetic resonance imaging during resting state. In this recording, EEG was recorded simultaneously. And so you see that in the two slices, there is a certain activity of the posterior visual cortex. And when you have the maximum activity in the occipital cortex, as revealed by bold, you have typically low amplitude alpha rims in the EEG. So in other words, when there is the reduction of alpha rims from scalp recorded EEG, this is typically related to a significant increase of occipital cortical areas. So the higher the cortical activity, the lower the amplitude of alpha rims. So the reduction of alpha rims reflects cortical activation. Ample alpha rims reflect the cortical inhibition. Again, a demonstration of the traffic light red cortical inhibition, ample alpha rims role. The other contribution of Fernando Lopez da Silva was related to the positive correlation between the bold signal from thalamic nuclei and alpha oscillations, alpha rims. The higher the alpha rims, the higher the bold signal in the thalamus. So alpha rims recorded from the scalp is related with both bold signal in the cerebral cortex and in the thalamus. This is another very interesting finding on the sources of resting state, eyes closed, alpha RIMS. In this case, you see the localization of the sources of resting state alpha RIMS. You see the cortical model, and in the upper graph, the green areas. In the lower graph, lower part of the graph, you see some red regions. These are the sources of two different components of alpha RIMS during resting state condition. The author were able to disentangle two components, two main principal components of recorded EEG activity by means of principal component analysis. And so they were able to distinguish between these two components and then use the separate components for the source localization of alpha RIMS. And so they could demonstrate that alpha RIMS is generated in the visual system, and particularly in the two main streams of the cortical visual system. The green areas are localized in the so-called dorsal visual stream, which is mainly parietal and occipital in the dorsal part of the cerebral cortex, and are typically related to visual spatial information processing and planning of reaching movements. So in the resting state, one part of alpha RIMS is related to the visual spatial attention, the modulation of visual spatial attention. In the lower part of the graph, the red regions are located in the so-called ventral visual stream, which is related to object recognition, related to classification of what we see when there are some visual stimuli. In this case, we are in the eyes closed condition, so no visuospatial information processing, no object recognition. But these results show that the alpha RIMS are related to the relaxation of dorsal visual stream and the relation of the green areas, and are related to relaxation, idling of the ventral visual stream, shown by the red regions in the cortical model. So the authors concluded that the scalp recorded EEG alpha RIMS in the resting state, eyes closed, are mainly related to the inhibition of visual cortical circuits, and that the dorsal and ventral visual streams are responsible of different components of EEG alpha RIMS. So we can conclude this part of the lesson emphasizing that the alpha RIMS are related to the synchronized activity of neurons, especially in the cortical and thalamic reciprocal circuits. And we know from the basic neurophysiology that these thalamocortical circuits are modulated by reticular ascending subcortical systems using dopamine, serotonin, and noradrenaline to modulate the oscillations of activity in the thalamic and cortical neurons. And so these are systems that regulate the cortical excitatory inhibitory balance and are responsible for the generation of EEG. In this picture, you see the results of an experiment by my research group testing the hypothesis that the cortical sources of resting state alpha RIMS reflect the cortical activity associated with the vigilance regulation. So if you have a change in alpha, this is related to a change in the vigilance, in the mental scanning of the environment or of the autobiographic memory. So the experiment was designed to have two recordings, two EEG recordings, one before one night of sleep deprivation, and the second EEG recording after one night of sleep deprivation. The results are shown in the picture, in which the vertical axis in each rectangle represents the activity of the cortical sources. The horizontal axis shows the frequency bands delta, theta, alpha 1, alpha 2, beta 1, beta 2, and gamma. And each rectangle shows the cortical regions of interest, the central, parietal, occipital, temporal, and limbic. In the graph, you see that there are the profile of cortical source activity from delta to gamma in blue for the EEG recording before the night of sleep deprivation. So it's a normal mental condition in the morning in healthy subjects. And you can see that there is a high cortical source activity in the alpha frequencies, in the parietal and occipital regions of interest. After one night of sleep deprivation, you have in the same subjects a resting state as closed condition again in the morning, but after one night of sleep deprivation. So with a sort of mental fog, a mental fatigue, a tiredness of the mind. So a lower vigilance in the same behavioral condition of resting state as closed. And indeed, you see that there is a decrease of resting state EEG activity in red. There is a clear reduction. And this means that if there is a decrease of vigilance after one night of sleep deprivation, this is associated with a reduction of alpha rhythms in the posterior areas. So this was a demonstration that the changes in alpha rhythms are related to changes in the vigilance during the resting state as closed condition. So we see that the alpha rhythms reflect the idling relaxation level of the visual cortical systems. And we wanted to test also this hypothesis. To do it, you see that we use a condition of in which we provided on the screen of a computer a sequence of visual stimuli, a visual task, which is represented in the right part of the monitor. So you see that at the beginning of an event, we showed a white circle in the left or right part of the screen. And this was the cue stimulus suggesting the site where the go stimulus, the green go stimulus, will appear after about 2 seconds. The cue stimulus was given at the sensory threshold level. The sensory threshold means that the duration of the cue stimulus was relatively short, so that the participant and a healthy subject was able to recognize the site of the stimulation about 50% of times. So in one event, he was able, for example, to say, okay, his cue was given in the right side. But in another event, in another trial, he was not able to do it, and so on. After The cue stimulus, we given immediately for 2 seconds a mask stimulus, avoiding that he was able to maintain the images in the retina of the stimulation. And then after the cue stimulus and the mask stimulus, we gave the go green circle in the right or left part of the stimulus. In the experiment that was inspired by the classical Posner's visual attention task, the cue stimulus was valid to predict the side of the go stimulus about 70% of cases. So a good probability of a correctness of the cue, the validity of the cue. For example, in this example is the 30% of cases in which the go stimulus was in the opposite part. The Posner task allowed us to verify that the patients, the participants, sorry, they were healthy subjects, were paid attention to the task, because we could replicate the typical case in which when the go stimulus in the same side of the cue, the participant is faster in the reaction time. When the go stimulus is in the opposite part of the cue stimulus, there is a delay, a cost in relation to an unexpected change of the side. After the participant responded with the pressing a button in relation to the go stimulus, if the go stimulus was given to the right side, the button to be pressed was on the right side. If the go stimulus was in the left side, the participant had to press the left button. And so after this task, this response, we asked to the participant, did you see the cue stimulus was in the left or right? And he replied left or right. And so we could measure if the participant was able to detect consciously the side of the cue stimulus. So we had the performance in terms of a visual primary consciousness of the cue stimulus, right or left, seen or unseen. We had the reaction time to demonstrate that the subject was following the task, and we had the simultaneous EEG recordings during the task and the possibility to analyze the alpha rims before the cue stimulus and during the task. And based on the verbal response, seen, not seen in relation to the cue stimulus, we could have two subsets of EEG epochs. The epochs, the events in which he was able to recognize the side of the cue stimulus, primary visual consciousness of the side, and the subset of epochs in which he was not able to detect the side of the stimulus. And so we could analyze separately the subset of seeing cue stimuli and not seeing cue stimuli, that we could compare the results. So you see that the results before the visual stimulation with the cue was represented in terms of cortical activation in the alpha frequency. And so you see that in the left column, we reported the results for not seeing cue stimuli, no visual primary consciousness, and in the red side, we reported the alpha source activity before the cue stimuli seeing. So we are showing the alpha source activity in the pre-stimulus period. And we see that the seeing cue stimuli were related to higher source activity in the dorsal stream of visual systems, in the occipital, posterior parietal dorsally, and in the dorsal premotor areas. So in other words, when there is a strong alpha source activity in the where visual stream in the occipital, parietal, and premotor areas, the person was able to recognize the cue stimulus. So the fluctuation of the vigilance during the pre-stimulus period was important to the visual primary consciousness when the cue stimulus was given. In another experiment with the same task, we wanted to test the hypothesis that if we give a perturbation magnetic stimulus on the parietal areas involved in the generation of alpha rims, before to give the cue stimulus, the subject was not able to recognize the cue stimulus when compared to the sham magnetic stimulation. So it's a fake magnetic stimulation, not true effective magnetic field, but just the sound to test the placebo effect. And so you see in the left part of the picture that we have a butterfly magnetic coil allowing to be very precise in the transcranial magnetic stimulation of the parietal cortex in the Brodmann area 7, in the posterior parietal cortex involved in visual and spatial information processing. And so we gave this train of magnetic stimulations in the parietal cortex to induce a perturbation of activity, a dysfunction of a parietal cortex for a while. After the transcranial magnetic stimulation in the parietal cortex, we gave the Q stimulus, the mask stimulus, and the GO stimulus. Like the previous experiment, but in this case, no EEG, but TMS perturbation of parietal cortex. And the results were reported on the right side of the picture. You see that we had three conditions: effective magnetic stimulation on the left parietal cortex in blue. In violet, the effective magnetic stimulation in the right parietal cortex. And in yellow, the sham condition, so no true magnetic stimulation, but just the sound. And in the horizontal line, we have the results of the experiment without any TMS stimulation, effective or sham, nothing, like in the EEG experiment. And the results show the number of seen Q stimuli. So the event, the cases in which the participant was able to say, yes, I saw the Q stimulus. It was in the left or right. In each session, in each condition, we gave 40 Q stimuli. So you see that the threshold of the Q stimulation was represented in the fact that without any valid or fake stimulation, we have about 20 Q stimuli seen. When there is the fake transcranial stimulation, the subject had the expected 50% of recognition of the Q stimulus. But when you gave the left or right effective magnetic stimulation in the parietal cortex, in the Brodmann area 7, we could see a small but significant reduction of the seeing Q stimulus detected consciously, visual primary consciousness. So we could conclude that the visual primary consciousness is due to different areas, and the perturbation of Brodmann area 7 in the parietal cortex is interfering in a small but significant way in the visual primary consciousness of a spatial feature of the cue stimulus, right or left. So this result in relation to the EEG result showed that the dorsal visual stream of visual spatial cortical neurons generates alpha rims in the resting state condition and can reflect the effectiveness of this system to recognize visual threshold stimuli. And that the perturbation of the parietal cortex, the posterior parietal cortex, can reduce our ability to have a conscious recognition of the visual stimuli for the spatial features. So we presented some evidence that the alpha rims reflected the vigilance level, that they are generated by a visual spatial stream and also of the object recognition stream of the visual system. And that this is casual, because if you have a perturbation, you can change the performance in the visual primary consciousness, and that cortical and thalamic neurons are interconnected each other functionally, and the generation of alpha rims. And we wanted to test this relation between resting state alpha rims and the consciousness level in a group of patients with a specific characteristic. They received a lesion in the brain over the tentorium. You see in the left side an encephalon, the tentorium line, upper the brain, in this case the right hemisphere, under the tentorium the cerebellum, and the brainstem. These patients received a lesion over the tentorium in the supratentorial regions. And this lesion was the cause of a very strong clinical manifestation. These patients were not able to interact with other people. They lost the consciousness in the sense that they were not aware of the environment, of the communications from other people, for the medical staff, the paramedical staff, the relatives, nothing. They seems not to have a consciousness of anything. However, they are awake. So the eyes are open, so they fix the wall. They do not react to any input. And then they fall asleep. So they have this cycle, wake and sleep, but they are not reactive to the environment. They do not have normal consciousness. And so the hypothesis was that in these patients, one would expect an abnormal resting state, alpha RIMS, during quiet wakefulness. And that this abnormality was related to the severity of the lesions, allowing the vigilance and the level of vigilance, level of consciousness. And so we recorded the resting state EEG in healthy subjects as a control group in the upper line, and in these patients with these clinical conditions called persistent vegetative state, because they resemble plants, unreactive to the verbal stimuli or visual stimuli and so on. So you see in the upper line, nose up, a model of the cerebral cortex with some red spots indicating the alpha source activity. And so the maximum is shown in the of source activity in alpha band. In the middle, we have a line of patients with persistent vegetative state. In the resting state, eyes closed. The patient was not able to respond to the instructions, so an experimenter closed the eyes manually. And so you can see that the main differences were in the source activity in the alpha band, a strong reduction of alpha RIMS in the middle line and also in the bottom line. So all the patients with persistent vegetative state show this drop of alpha RIMS, possibly related to the abnormalities in the level of vigilance and consciousness. But the two lines show two different subgroups of patients. In the middle line, we have patients that will recover a certain degree of level of consciousness after 3 months. And so we call these patients who will recover the level of vigilance and consciousness at the follow-up. But during the resting state EEG recordings, the patients of the middle line or bottom line did not have any consciousness of the environment. And in the bottom line, you see the patients who will not recover the level of consciousness after 3 months show even more reduction of alpha source activity in the resting state, eyes closed. So we could conclude that patients with a very abnormal level of consciousness and vigilance are associated with disruption of resting state alpha RIMS. But the patients who will recover the level of vigilance after 3 months show a less disruption of resting state alpha RIMS. Another opportunity to investigate the relation between alpha RIMS and the level of vigilance was given by the COVID-19. Because you know that in the COVID-19, you had some people who were recovered in the hospital for respiratory distress. They experienced a severe health condition for some periods. But fortunately, some of them were able to recover. They became negative to the virus. However, some of them, after 3 or 4, even 6 months, even 1 year, claim that they suffer from mental fog, some mental fatigue, a chronic mental fatigue, some general difficulties in concentrating. And so they are patients with exactly the kind of conditions of interest to test the hypothesis that alpha RIMS is related to vigilance level. So in these patients with post-COVID mental fog, we recorded EEG in the resting state as closed condition. And we compared these patients with the post-COVID mental fog with subjects who didn't take the COVID in the graph, you see the alpha source activity in frontal, central, parietal, and so on. And in blue, you have the unaffected healthy subjects. And the post-COVID patients with mental fog were divided in 2 subgroups. The subgroup with 3 or more mental fog symptoms based on a scale of on mental fog, mental fatigue, difficulties in concentrating, and so on. And so these post-COVID patients with 3 or more symptoms are in red. The patients with 1 or 2 symptoms in the scale of mental fog are reported in green. So they are mild post-COVID patients. And you see that there is a very clear drop of alpha source activity in the post-COVID patients with 3 or more brain fog, mental fog symptoms when compared to healthy subjects unaffected by COVID and the patients with 1 or 2 symptoms of mental fog. So this was another demonstration that patients with this mental fatigue, mental fog, with this disturbance of a level of vigilance consciousness show some abnormal reduction of alpha rhythms. In the last part of this talk, I will talk about the level of vigilance in patients with neurodegenerative diseases during the aging. The diseases such as Alzheimer's disease or Parkinson's disease, which affect the brain cognitive systems underpinning episodic memory or attention or executive functions. And so I will report some findings of a consortium, an international consortium called PD Waves. And the PD Waves consortium was formed by key researchers of Italian centers for the care and the investigations on dementia, but also key researchers from European countries, also Asiatic key researchers and institutions. You see an overview of these key researchers and their institutions in this international consortium. I am the leader of this consortium. I founded about 10 years ago. And we collected the data together and we discussed the results and published the papers. The focus of the PD Waves consortium was understanding the vigilance dysfunctions as revealed by EEG activity in patients with cognitive deficits due to Alzheimer's disease, Parkinson's disease, and other neurodegenerative diseases. The most severe form of cognitive deficits are classified as dementia. Dementia means severe cognitive deficits, memory, frontal executive functions, or visuospatial skills, language, for example. And when these cognitive deficits affect the activities of daily living. So the older people with dementia are not able to wear oneself or to prepare a meal, to do shopping, to acquire some important goods. They are not able to use instruments. They are not able to use the remote controller of TV or to use a machine to obtain the tickets to get to the bus and so on. So they need a caregiver for all these activities. And dementia, like the fever, is a symptom. And the causes of this symptom can be very different. Our interest is in the neurodegenerative diseases. These are diseases which are related to the accumulation of neuropathology in the brain. And this neuropathology can be localized in different places in the different diseases. And this neuropathology can be related to different proteins in different neurodegenerative diseases. Here we have two cases, the case of Alzheimer's disease and Parkinson's and the Lewy body diseases. In the left, Alzheimer's disease is represented as affected by two proteins, two pathological proteins, toxic proteins for the neurons. In brown is the tau protein, which is a protein that is important for synaptic transmission and also to take the microtubules of the axons of the neurons in a stable and effective way. When this protein is phosphorylized, is changed the shape and show dysfunctions. It's really impactful because the accumulation of tau induces the death of the neuron and the neurodegeneration. The other protein responsible for Alzheimer's disease is the amyloid beta 42. And you see in blue that there is a large area in the frontal and a large area in the posterior cerebral cortex showing this accumulation of amyloid beta 42. And you see that there is an overlapping with the regions showing the accumulation of tau, and this interaction between amyloid beta 42 and tau seems to be the trigger of the neurodegeneration, the neural loss, synapses loss, and severe cognitive dysfunctions. The typical manifestation of amyloid beta 42 accumulation is in the plaques outside the neurons, a sort of mass of amyloid extracellularly. The phosphorylized tau protein is the core of neurofibrillary tangles within the neurons affecting the neural transmission and inducing the death of the neurons. One particular aspect of the picture is that you can find this tau abnormal accumulation, not only in the cerebral cortex, and you see the regions of mesial temporal cortex and the hypothalamus and amygdala, which are very important in the episodic memory, but not only in the brain, but also in the brainstem. And you see here a spot of tauopathy in Alzheimer's disease in the brain, in forebrain, in the brainstem, and this means that the subcortical neuromodulatory ascending systems regulating thalamocortical interaction, the cortical excitation, and the vigilance are impacted by Alzheimer's disease. On the right side, we have the accumulation of neuropathology in Parkinson-Levy-Body disease. You see that the neuropathology is related to the so-called Levy bodies, which are these mass of proteins within the neurons with a core toxic protein called alpha-synuclein. Alpha-synuclein is a protein also involved in the synapses and neural transmission. And when this mass of proteins appears within the neurons, they unfortunately die. And you see that you have brainstem Levy bodies, a lot of them, so impacting sleep and wakefulness systems, but also cortical Levy bodies responsible for the cognitive deficits. So there are differences in locations or the kind of proteins, but the final result is that we have vigilance, dysfunctions, and cognitive deficits at a given point. Alzheimer's disease are the most prevalent disease belonging to dementia. And you see that in the evolution of the disease, we have some, for example, Alzheimer's disease. In brown, you have tauopathy. In blue, amyloid beta 42 accumulation. You see that in the history, in the natural history of the Alzheimer's disease, the tauopathy appears in advance, even when you don't have the clinical symptoms, no cognitive deficits. And then you have an accumulation of amyloid beta and interaction with tauopathy, and the manifestation of mild cognitive impairment, in which you can measure some decline of memory or other functions, but the people are autonomous, are independent, can drive the car, they can control for the children and can do shopping and so on. So the mild cognitive impairment is compatible with an autonomous life. But when there is an increase in the accumulation of pathology, the cognitive deficits become more severe. And so when there is the disabilities in the activities of daily living, there is the diagnosis of dementia, in this case due to Alzheimer's disease, in other cases due to Parkinson's disease or Levy-Body disease and so on. The National Institute of Aging in United States and the Alzheimer's associations published these clinical guidelines as a framework to the use of biomarkers for the neurobiological diagnosis of Alzheimer's disease. And you see that the amyloidosis in the brain can be revealed by doses of a beta 42 in the cerebrospinal fluid, or with some binding of ligands of amyloid by positron emission tomography, in this case with the ligand called fluor beta peer. Along the same line, one can measure an abnormal dose of tau and phosphotau in the cerebrospinal fluid, or can see the binding of tau in the brain with positron emission tomography. So in the framework, they called A as amyloid measures and tau, T as tau-T measures, the biomarkers allowing the diagnosis of Alzheimer's disease. They also suggest to use magnetic resonance imaging, the volumetric measure of eventual atrophy, and the use of FDG PET, the metabolic PET, in the brain to establish the severity of the brain disease due to Alzheimer's. And then the evaluation of cognitive deficit with clinical scales and neuropsychological tests, pencil and papers, and also some computerized cognitive battery. You see from this clinical guideline that the level of consciousness and sleep disturbances are not considered as relevant for the clinical diagnosis of Alzheimer's disease. This is something really wrong, because the patients with Alzheimer's disease suffer from mental fog and also exaggerated naps, exaggerated sleep during daytime. So we wanted to demonstrate that Alzheimer's disease patients with a diagnosis based on cerebrospinal fluid measures of amyloid and tau suffer from vigilance dysfunctions. And so we enrolled patients with mild cognitive impairment due to Alzheimer's disease based on cerebrospinal fluid biomarkers, and compare the EEG results with a group of patients with mild cognitive impairment not due to Alzheimer's disease. And so we show that the group of Alzheimer's disease mild cognitive impairment in the red show lower alpha source activity when compared to non-Alzheimer's disease patients with mild cognitive impairment. So this was a very clear demonstration that the Alzheimer's disease patients with a diagnosis related to biomarkers from CSF show the vigilance dysfunctions, and as revealed with poor resting state alpha RIMS. Another experiment which is relevant for understanding the basis of vigilance dysfunctions in Alzheimer's disease patients, tested the integrity of cholinergic tracts from basal forebrain to cerebral cortex. We know that the cholinergic system is affected in Alzheimer's disease, and the most diffuse treatment is based on acetylcholinesterase inhibitors enhancing the cholinergic transmission. Because we know that Alzheimer's disease neuropathology, amyloid and tau and phospho-tau affect particularly cholinergic neurons. And today it's possible to quantify the lesions in the cholinergic tract from basal forebrain to cerebral cortex, because we have a mask in the magnetic resonance imaging indicating where the cholinergic tracts are in the brain. And you see here the tracts in red, yellow, and green. And so we measure the lesions in these tracts. We recorded EEG in Alzheimer's disease patients with cognitive impairment. We measure these lesions in the cholinergic tracts, and we divided the Alzheimer's MCI patients in those with less cholinergic lesions and those with more cholinergic lesions. So you see here the results. When compared to normal elderly subjects, in the upper line, the patients with mild cognitive impairment due to Alzheimer's disease with less cholinergic lesions, C minus, show a drop of Alzheimer's disease compared to normal subjects, but still evident in the posterior areas. And patients with mild cognitive impairment and particular lesions in the cholinergic tracts show a greater drop of alpha source activity in the resting state, eyes closed. So at least at the group level, the integrity of relatively preserved cholinergic system is related to higher alpha source activity in the resting state, eyes closed. So another demonstration that the vigilance systems related to cholinergic modulation of thalamocortical activity are relevant in Alzheimer's disease patients, even in the mild cognitive impairment state. And we confirm this relevance of cholinergic system to maintain the vigilance level with another experiment. And this is the last slide today. And in this experiment, we enrolled Alzheimer's disease patients with dementia, mild to moderate dementia. And we recorded resting state, eyes closed EEG before to start with cholinergic therapy. Then we gave the treatment, the cholinergic therapy, we acetylcholinesterase inhibitors, donepezil, for 1 year. And then we recorded again the resting state EEG in eyes closed. And we measured before the treatment and after 1 year of the treatment, the cognitive level with mini-mental state evaluation test. In this graph, we represented the EEG source activity in the second recording in the follow-up minus the first recording before starting the treatment. So when you see 0, this means that the source activity didn't change after 1 year of treatment. If you see negative values under the blue line, this means that there is a decrease of source activity in a given frequency after 1 year. The results were presented in two subgroups of Alzheimer's disease dementia patients, those who didn't respond to the therapy, didn't show cognitive improvement or stabilization after 1 year in red. And you see that the red population of non-responders to cholinergic therapy show a very dramatic reduction of alpha at the follow-up, very big negative values of the difference. And especially in the occipital and temporal regions of interest for the source estimation. The group of people responding to the cholinergic therapy, so they had the same mini-mental state evaluation score after 1 year of treatment, or even a small gain in blue, show, unfortunately, a reduction of alpha RIMS after 1 year, even with a stable cognitive status. And this shows that even if the therapy provides some beneficial effect at the cognitive level, the vigilance systems show a decline, an impairment increased after 1 year, even with a good response to the cholinergic therapy. And this use of the cholinergic therapy and the relation with EEG was another demonstration of the importance of cholinergic transmission and connectivity in the function of vigilance in quiet wakefulness tested with the resting state eyes closed paradigm. So the talk is ended today.